Hi, everyone. Thank you for joining us today for our second webinar in a series of BioCord-based webinars. Today, we will be focusing on year-round compliance and how to achieve stringent treatment limits in cold conditions. So this is the second webinar in a series that Bishop Water will be hosting over the next few weeks. These BioCord-based webinars will explore current topics in wastewater treatment. Last week, we introduced the BioCord reactor and gave an overview of how it works and its applications. This week, we're focusing on cold weather removals using BioCord, and next week, we'll be talking about how to get the most out of your pilot project. So for outlines of these topics and to see a schedule of our webinars, you can just look us up or follow us on Twitter or LinkedIn. You can also get the information there and find the link to register for our webinars, which will be occurring Thursdays at 2 p.m. EST. So these webinars will be presented by myself, Christine Gann, and my colleague, Roberta Maffatone. We are both wastewater and engineering specialists here at Bishop, and we can be contacted via email if you have any questions about the content of these webinars or about these topics in general. I do want to mention a few housekeeping items before we get started. First, we will be recording the webinar, so we ask that everyone please keep their microphones on mute if you haven't already done so. As well, if you would like to ask a question, please just use the chat function. You can access this by clicking on the chat icon on the top right corner of your screen, and then type your question in the dialog box, which will appear down near the bottom. For those of you that have joined by phone, you're welcome to email your questions to christine@bishopwater.ca. I won't be addressing questions as they come in, but I will try to answer all of them at the end of the webinar, just to keep us on schedule. So if there happen to be more questions that I can answer during the Q&A session, I will address them by email and send the responses to everyone who is registered. We'll also follow up this webinar with an email that has a link to a recorded webinar and some additional information about the BioCord reactors as well. So to begin, I always like to start off with a summary of some basic concepts to warm everybody up and give you a little bit of context as to what's to come. So today I'm talking about before cold weather treatment. I want to go over some concepts in biological wastewater treatment. So we do use microorganisms to remove contaminants. Microorganisms can remove organic matter and nutrients from wastewater by participating in chemical reactions. And this is important for cold weather treatment. So for example, organic matter is one of the main contaminants in wastewater. It consists of all carbon-based compounds. And its presence in wastewater comes from things like food particles, human waste products, leaf or plant matter, and biomass, amongst other things. As shown, organic carbon is removed by aerobic or oxygen-requiring microorganisms via the chemical reactions that I have shown here. Heterotrophic means that the bacteria need organic carbon from an external source to use as food and to grow and live. So this is just a simplified equation here that basically outlines what all the components are that are involved in the reduction of organic carbon. So the microorganisms are using oxygen, or O2 here, to convert the organic carbon into what is essentially energy, carbon dioxide, and water, as well as some residual elements, thereby eliminating organic carbon that's present in wastewater. And then ultimately, the energy that's produced is used for the growth and proliferation so that more microorganisms are able to, the energy is used to grow new microorganisms so that even more organic carbon can be reduced, and so on until you have a biological treatment system. Another example is the chemical reactions that are used to remove ammonia, which is a key nutrient contaminant in wastewater along with phosphorus. So ammonia is largely removed by aerobic, so again, oxygen-loving or oxygen-requiring autotrophic microorganisms. So in this case, autotrophic just means that they don't need organic carbon for food. So they can produce their own energy from inorganic substances such as ammonia. In this example, we can see that nitrifiers use oxygen to eliminate ammonia by converting it to nitrate and then to nitrate and so on. And of course, this diagram and these equations are massively simplified. In natural systems, there is a lot more going on than just this to remove ammonia, but I just wanted to illustrate kind of two main examples of chemical reactions in wastewater that are relevant to us. So even if microorganisms are present, which they typically are in all wastewaters, these chemical reactions are not always a given. So they're not always happening, and even when they are happening, they're not always happening quickly. If you took high school chemistry or biology, you may recall that the rate at which these chemical reactions occur, or what we call the kinetics of the reaction, can depend on a huge number of different factors. And these factors all influence and interact with one another as well, which can definitely complicate things if you don't have a robust enough system. So some parameters or conditions that can influence reaction rate include the growth rate of the specific microbe. So like humans, some bacteria just metabolize substrates faster than others. Notoriously, heterotrophs are faster growing than autotrophs. So BOD consuming bacteria will grow faster than ammonia consuming bacteria. And this is important to note, but I'll come back to this in a bit. The concentration of substrates and products in the reaction also matters. So having a high concentration of substrates or reactants, such as organic carbon or ammonia, tends to drive the reaction forward and make it go faster. Although this also depends on things like the presence of inhibitors and other reactions, oxygen concentration, and nutrient deficiencies, as things like nutrient deficiencies can slow down your reactions as well. Reaction rates can also be influenced by other environmental conditions such as pH level, redox potential, conductivity, and dissolved oxygen, and most importantly, temperature. So this is where cold temperatures come into play when you're considering cold weather treatment and improving your ammonia removals in the wintertime. So the environmental temperature, or the water temperature that your microorganisms are in, has one of the most significant effects on microbial reaction rates. We can represent this concept using this modified Arrhenius equation. And I don't want to get into too many details about this equation, but I did want to include it because it really nicely represents the relationship between K, which is the rate constant that determines how fast the reaction goes, and temperature, which is capital T. And so these other parameters in the equation, so K20 and theta here, are constants that change depending on the type of system that you have. But no matter what the value of these two other parameters are, the trend stays the same. So as you increase temperature, the reaction rate will also increase, and vice versa. That relationship will always stay the same, up to a maximum temperature, of course, after which the activity will decrease again. So yeah, you can see this represented by this graph to the right, which plots temperature on the x-axis against the percent maximum activity of the microorganism on the y-axis. I've also seen graphs like this where the y-axis is the growth rate or number of cell divisions rather than percent maximum activity. It means the same thing, and the curve will still look like this approximately. Keep in mind that this is a generalized plot using standard values. So basically where the peak of this curve occurs, or where the optimal temperature range is for your microorganism, it will vary based on, of course, the type of microorganism that you're looking at, your system, and environmental conditions. But I'm going to reiterate it, the trend will remain the same. So as you can see, temperature does pretty significantly influence the activity of microorganisms and the rate of reaction. So you can see at standard conditions, when you reach around 15 degrees Celsius, you can see that activity has already reduced by up to 80% in a generalized standard conditions case. And that typically demarcates what we would consider cold weather conditions and warm weather conditions. When your water temperature is below 15 degrees Celsius, you can typically consider those conditions to be cold weather. So like I mentioned, the reactions that each microorganism participates in will have slightly different kinetics from one another. A very important distinction is between heterotrophic bacteria that utilize organic carbon and the autotrophic bacteria that utilize ammonia. or nitrifiers. So nitrifiers that reduce ammonia are actually more sensitive to cold temperatures than heterotrophs. So the curve for nitrifiers is flattened a bit, with the influence of colder temperatures on activity being a bit more apparent. And on top of that, nitrifiers are slower growing overall and take longer to complete their reactions, which means that if there is a reactant that both nitrifiers and heterotrophic bacteria need, such as oxygen, and there's not enough of it to go around, the nitrifiers will be pretty easily outcompeted and die off because they can't complete their reactions for growth and proliferation. So this is why it's super hard to get good nitrification in conventional biological treatment systems. And it's also why achieving ammonia reductions in the wintertime is so difficult. It's like a double whammy of both slow metabolism and sensitivity to the cold. So what can you do to combat this and enhance cold weather nitrification or just increase cold weather treatment in general? Well, you can try to increase the reaction rate of the bacteria as much as possible. So you can try a different couple approaches to do this. You can really try to focus on the temperature aspect of microbial kinetics by adding heat to the system or preventing heat loss from the system. Or you can also try to optimize other factors that may contribute to increasing those reaction rates and just overall getting better reductions and better activity. So let's explore. The most obvious and effective solution would be to add heat into your system to maintain a warm enough environment for your bacteria. But this is usually pretty expensive and energy intensive, especially if your existing system is quite large or is an open system. So it's really not the most practical option, although there are ways to do it a little bit more efficiently, and I'll touch on that near the end of this webinar. You could also try and prevent heat loss from the system. So depending on your facility, the influent wastewater may stay quite warm, or at least not cold, even in the wintertime. And the majority of heat loss actually happens within the biological treatment system, which is usually a large open aerated basin or pond. So you can use things like floating covers or insulated tanks to retain as much heat as possible, as well as retain the microbial activity. Or you can mitigate against this heat loss using BioCord reactors, which use biofilms for enhanced wastewater treatment. And I'll explain exactly how that works in just a little bit. Lastly, we can also increase cold weather treatment by optimizing other factors that influence the reaction rate of microbes, or specifically nitrifiers, that aren't just related to temperature. And again, we can use our BioCord reactors to do this in a number of ways, which I'll be explaining. So we're at a good point now to just give a brief overview on the BioCord reactors and what they are, for those who were not able to attend last week's webinar, and then we'll get into how it can directly address cold weather treatment. So the BioCord reactor is a bioreactor developed by Bishop Water Technologies. It provides wastewater treating bacteria with the optimal conditions for growth and proliferation to allow for high levels of microbial activity. So this includes the provision of a string-like media that has a high surface area and charge for the microbes to attach onto, as well as a unique aeration system with an enhanced oxygen transfer capability to supply lots of oxygen directly to these microbes. And as seen on this diagram to the right, providing these things for the microbes leads to the formation of a really robust biofilm, which is essentially a consortium of a lot of microorganisms that stick to a surface, as well as to each other in really, really high numbers. BioCord reactors can be used in municipal, industrial, and agricultural applications to enhance the removals of BOD, COD, TSS, as well as nitrogen. It is a modular and customizable technology that's designed specifically for each individual application. As you can see, it can be installed in situ within a lagoon or basin, and it can also be used to enhance or improve treatment reductions and increase capacity at an existing facility. Alternatively, it can be designed as a standalone or a side stream system if required. So now let's get into it. How exactly does BioCord increase cold weather nitrification? So BioCord reactors can help combat cold weather effects by doing two things. as mentioned, it addresses the temperature issue directly by insulating the microorganisms from the cold using its biofilm properties. And second, it allows for the optimization of other parameters that influence or can help to speed up microbial reaction rates. So the biofilm that develops on BioCord provides an insulating effect for the microorganisms. Because there are so many more microbes present in a biofilm than in a suspended sludge system, and because they're all stuck together and concentrated within this biofilm, each individual bacteria feels the effects of lowered temperatures less. So just by nature of having a biofilm system, you can already start to mitigate against cold temperature effects. Um, in addition, the biofilm develops in layers. I touched on this briefly last week. So different microclimates can exist within a single biofilm, as shown in this diagram here. So as you can see, different substrates such as CO2, oxygen, soluble organics, nutrients, and oxygen can diffuse in and out of the biofilm at different rates. So each layer within a biofilm will have varying levels of components, and each layer will also have different environmental temperatures. So the inner layers of a biofilm will be a lot more insulated from cold water temperatures, which allows for continuous reductions even as water temperatures at the biofilm liquid boundary layer are lowered. So yes, this is all happening at a micro scale, which you may not believe is very significant, but it truly does make a difference. And I have some case studies to share with you in a bit that will show you how. So here's another diagram that I really like that illustrates this concept. So as you can see here, if you foster a robust biofilm, you can get both heterotrophic and nitrifying bacteria coexisting within a single biofilm. So not only is your embedded nitrifying bacteria insulated against the cold, but you're also able to reduce the competition with heterotrophs because the bacteria are fixed in place. So nitrifiers will have the time and space to grow separately, even though they're growing all within this biofilm together. And then as well, you can get both aerobic and anaerobic microorganisms developing within a single biofilm, which can be helpful if your goal is something like removing total nitrogen, which requires both. Another thing that I wanted to bring up is that not all biofilms are the same. Yes, conceptually, biofilm systems tend to work on the same principles, but systems are built and operate differently, and that can have sometimes huge impacts on the way the biofilm develops and performs. For example, different systems will foster biofilms with different thicknesses, resiliencies, boundary layers, diffusion rates of substrates and gases into and out of the biofilm, and attachment and detachment coefficients for the microbes themselves. So one big difference between BioCord and other similar technologies is that the media carrier for the biofilm in a BioCord reactor is fixed in place, so it's static, rather than having to be kept suspended or moving within a basin, such as with MBBRs or RBCs. So not only is this more energy efficient, but it also allows the biofilm on a BioCord reactor to grow a little bit thicker, as can be seen from these images and active biomass measurements here on the left. And so that helps to better maintain this cold weather insulating effect. So in literature, this relationship can be described as follows. As thickness of the active biofilm layer increases, the sensitivity of the process to temperature variations is significantly reduced. As well, I do want to say we do actually have a BioCord model that we use in wastewater modeling programs, such as GPSX, and that takes into account all these operational and empirical parameters that are specific to BioCord. And we can use this to design and size our systems using process modeling, as well as for verification purposes to predict what kind of effluent quality we're going to get based on the modeling. And so this BioCord model is something that's continuously being refined and made even better as we implement more projects and get more and more data. But I'm going to leave this here because Roberta will be speaking a bit more about this in the process optimization webinar that she'll be hosting. So don't forget to register for that one if you haven't already. It's going to be really good. So now we can talk about our first research study, which we carried out in collaboration with Windsor University to analyze the microbial community dynamics of a BioCord reactor system in an open lagoon setting. So the broad objective of this study was to look at what, if any, environmental factors most impact the growth and activity of nitrifiers in BioCord. And specifically as it relates to cold weather treatment, we wanted to see if we could verify that the activity of nitrifiers on BioCord was retained, even as temperatures decreased to below 15 degrees Celsius. So our BioCord reactor system was installed in August of 2016 in lagoon number 4 of a 5-cell facility located in Dundalk, Ontario. We installed our system and allowed it to reach steady state, which it took about 8 weeks, and then we began to collect biofilm samples from the BioCord reactors that were within the lagoon. So we first took biofilm samples in warm weather conditions, which in the context of biological treatment, I said earlier, is defined as water temperatures above 15 degrees Celsius. We then preserved those samples and waited until the water temperatures dropped to consistently below 15 degrees Celsius, and then again took biofilm samples from those reactors. And then we did in-lab extractions of RNA and DNA from the biofilm so that we could look at the active microbial communities and identify the key members of the microbial communities. And because we also wanted to look at the impact of location as well as temperature on nitrifier activity, we then moved the BioCord reactor system from Lagoon 4 into Lagoon 2 the following year and did the same biofilm sampling and analysis for warm weather and cold weather conditions. So all in all, we had 4 total biofilm samples. So here are the lab taxonomy results, which show the relative abundances of the active and total nitrifying communities that developed on the BioCord. So the results from when the BioCord system was in Lagoon 4 are shown on the left-hand side of this black bar, and the results from when the system was in Lagoon 2 are shown on the right-hand side. And the water temperature at the time of sampling is shown on the top axis here. So from this taxonomy graph, you can see that the abundances of nitrifiers were impacted where the reactors were located, but they weren't as impacted by temperature. So although you can see an apparent difference in the active nitrifiers going from Lagoon 4 to Lagoon 2, within the same lagoon, there was no significant change in nitrifying genera as the water temperatures decreased from warm weather to cold weather conditions, which means that compared to suspended growth processes, the microbial community can continue to maintain activity even in cold water temperatures. And as well, although you can see a slight decrease in the abundance of active nitrifiers from Lagoon 2, this decrease was not actually statistically significant. So statistically, there wasn't too much of an impact. The other interesting thing that we found was that Candidatus nitritoga was actually the dominant nitrifier species identified in the lab taxonomy based on field samples. So Candidatus nitritoga encodes genes that allow it to survive and proliferate during the depletion of reactants, meaning that it's suitable for dynamic environments such as lagoons or open systems that are exposed to the elements. Previous research has also shown that Candidatus nitritoga is the most well-adapted nitrite oxidizer to colder temperatures, which could definitely explain its dominance in this cold temperature environment as well. So that's just a little extra tidbit, a little bit of information that we were able to gather about BioCord's microbial community as it relates to cold weather treatment that we found really interesting. We used a canonical correspondence analysis, or a CCA correlation chart, to summarize all of our results. Since we weren't just looking at the influence of temperature and location, we were actually also looking at the influence of all the other key factors like pH, TSS, all the different parameters that you would find on wastewater. So CCA is just a statistics method that's used to correlate parameters with one another, so you can determine what parameter has the most impact on something else. So in this case, what parameter has the most impact on nitrifier growth and activity. So you don't have to worry about decoding or reading this graph, but I will point out a few key concepts on here. So each point or each dot on this graph represents a sample. And you can see that the communities between Lagoon 2 and Lagoon 4 are plotted quite far away from each other on this graph, which indicates distinct microbial communities were developed based on where we installed the system. And when you look at this graph, you can also see that all of the nitrogen parameters are increasing together on this x-axis, which indicates that nitrogen was actually the most significant driver or in the difference between communities, so nitrogen concentration. So one of the main takeaways from this study was that the concentration of nitrogen in your influent stream or the location of your installation does have an impact on your treatment. So it's important to make sure that you're picking an optimal installation location when you're upgrading your system using a technology or process. And that's something that we help with when designing our BioCord systems. We look at all the data available and then choose based on this where the best spot for treatment or for an installation is. And secondly, and most importantly to us to help verify our hypothesis, is that you can also see that variation due to temperature does not or does have a lot of overlap where nitrogen has none. And that means that temperature was not a significant driver of nitrifier variation or nitrifier activity variation between samples, and that the activity of nitrifiers was not significantly impacted by water temperature increases or decreases, sorry. So now coming back to the main points of how BioCord Reactor increases cold weather nitrification, we just spoke about how they can help to insulate the biofilm from cold temperatures, leading to sustained nitrifier activity. Now we can focus on how BioCord optimizes some other factors that can influence how fast they reduce ammonia. One of the main ways BioCord does this is by increasing overall concentration of nitrifiers present. This is a concept that I've covered before, but I really wanted to reiterate it here because this temperature effect can really sometimes trip people up. They think that as soon as the water temperature gets below 15 degrees Celsius or 10 degrees Celsius and your curve is all the way down here, biological treatment pretty much just stops and you don't get treatment. And in order to get good nitrification, you need to increase that temperature or heat up the system, but that's just not true. From what we saw, number one, BioCord has an insulating effect that can actually help to mitigate against this curve and this effect of cold water temperatures. But even if not, so even if you do get slowed activity at colder temperatures, that doesn't mean you're not going to get treatment. If you have more nitrifiers present, then you get more total reactions happening, which in general leads to better ammonia reductions. So BioCord can help to increase the number of active nitrifiers present in a couple of different ways. As mentioned before, one way is by minimizing the competition with heterotrophs. Since each bacteria is localized to a specific part of the BioCord, the nitrifiers won't be outcompeted. The second way is by ensuring an abundance of dissolved oxygen, which I'm going to be talking about next, and also by preventing washout of nitrifiers or increasing sludge age. So let's talk about oxygen. If you recall our equations from earlier, you can see that both heterotrophs and nitrifiers require dissolved oxygen, or O2, as a substrate to complete their reactions and reduce either BOD or ammonia. So without oxygen or without dissolved oxygen, the reaction just isn't going to happen. It's extremely important, and it's often the limiting factor in biological treatment. As you can see, since full nitrification is a two-step process where both processes require oxygen, the process of reducing ammonia actually requires more oxygen than the process of reducing organic carbon does. So in cold weather conditions, it's really important to make sure that these oxygen requirements are consistently being met, or you're really not doing your system any favors, and it can really slow down your treatment rate. So to ensure that dissolved oxygen is in abundance, we designed our BioCord systems to have an integrated aeration system with a very high oxygen transfer efficiency. So oxygen molecules have to first be dissolved in water before they can actually be used, or else you're just wasting energy putting air into your system that's not dissolving, it's not becoming soluble, so it can't be uptaken by the microorganisms. So if you have a high OTE system using smaller fine bubbles like the one that we use, this ensures that you're going to be getting better performance with less energy and cost. And since these diffusers are placed right underneath our biofilm, we can ensure that our DO is being delivered directly to the biofilm that needs it the most, and that it's constantly being replenished. And this in turn helps to maintain the activity of our nitrifiers in cold weather. If you're relying only on a suspended sludge system, putting in a lot of DO or using a really efficient aeration system isn't enough to really improve cold weather nitrification. And that's because nitrifiers are slow growing, as I've mentioned, and will get washed out by the hydraulic flow. So even if you put a ton of dissolved oxygen into your system, there just won't be enough bacteria actually in it to make a significant impact on nitrification. So again, BioCord uses biofilm to prevent this nitrifier washout and increase the amount of time your bacteria can stay in your system. So this time spent in the system by the nitrifiers is called sludge age, or solids retention time. And if you increase your sludge age, you can help decrease the influence of cold temperatures on your microorganisms. This is also something that's very well documented in literature. So of course, there's also the reactor design or operational factors that can help to influence cold weather nitrification and improve it. One of the advantages with Bishop is that we design our BioCord reactor systems for each site-specific application. Because reaction rates are heavily influenced by the concentration of reactants and products. This also means that if you don't have enough ammonia or BOD for the amount of bacteria that you plan on developing or have developed, then the activity of your nitrifiers overall will be slowed. So this is why we design our systems to have just the right amount of surface area or biofilm development and oxygen to optimize this food to microorganism ratio, or F to M ratio. So that we're just in the right sweet spot where your balance of heterotrophs, nitrifiers, and food are all optimized to achieve the best removals you can at your specific facility and for cold weather conditions. And we can do this because we've done enough work with BioCord that we've discerned things like how much ammonia can be removed per meter of BioCord media, its oxygen uptake rate, and things like that. And like I mentioned previously, we have developed a model for BioCord that we use to help us design our systems, and that will be discussed in our process optimization webinar. Also, I would suggest our piloting webinar that's taking place next week if you're interested in learning a little bit more about what the most important parameters are for you to be collecting at your facility to optimize the technology design and how you can do that during a pilot project. Of course, your ammonia levels come in, or your F to M ratio won't always be consistent. You'll always get fluctuations depending on what your flow rate is, the time of year, and all of that. But BioCord systems are very robust. So as long as we're designing for an average or most frequent conditions, we can still achieve really high reaction rates and nitrification despite these variations. We can also design BioCord systems with different customization options to ensure we're maxing out our cold weather nitrification as much as possible. So if flows are highly variable and prone to messing with that F to M ratio, we can do things like install baffles to maximize contact time or increase the overall HRT. And that's to ensure that the nitrifiers have time to complete their reactions. Or if there are other limiting factors in your wastewater, such as alkalinity, which is required for nitrification, we can also supplement that to our BioCord reactors using like automatic dosing systems and things like that, just to maintain those optimal conditions. We also have other options for increasing heat retention and temperatures as well. For example, BioCord reactor systems can come in containerized or side stream versions, which can be insulated and heated, and that helps to increase temperatures without expending too much extra energy, since you're just heating kind of a smaller system. We also have options such as floating covers, like the ones that I mentioned before, which can be rolled out in the winter to prevent heat loss. I'll now present a couple of cold weather case studies that help demonstrate BioCord's wintertime nitrification capabilities. I'll be reviewing two cold weather BioCord projects. One is from a lagoon facility in St. Henry, Ohio, and the second will review our results from a pilot project treating more industrial effluent. So I'll first talk about St. Henry. St. Henry is a 4-cell lagoon to treat wastewater from the surrounding community, as well as effluent wastewater from a turkey processing plant nearby. The village had difficulties maintaining ammonia removals during winter operations, despite having quite a robust aeration system in their first cell. So they wanted to assess different technology options for treatment upgrades to help the facility meet their wintertime ammonia discharge limits. So this is how we set up our pilot. We installed a side stream unit between cells 2 and 3, and we sized our reactor based on the historical data that they provided to us. So we designed our pilot system for an influent ammonia of around 15 milligrams per liter and an influent BOD of around 75 milligrams per liter. And we set our flow rate to achieve a 12-hour HRT in the system, which would match the HRT of the full-scale design that we had put together. And then we monitored all of our parameters, or all of our key parameters, from the inlet and the outlet of the system weekly from October 2018 to May 2018. Or sorry, October 2017 to May 2018. So here are the results for BOD and ammonia that we achieved. On these graphs, the red line represents the influent concentration. The blue line represents the concentration of the BioCord effluent. And then the light gray line in the background is the temperature of the water entering our BioCord system, which is plotted on the secondary axis. So you can see between the end of December through about March, water temperatures were pretty consistently less than 5 degrees Celsius, sometimes reaching as low as 0.1 or 0.2 degrees Celsius. So yeah, I do want to point out first and foremost, I hope you can see it on this graph, but I did want to point out that the actual influent concentrations of BOD and ammonia entering the system were significantly higher than the design parameters. I've shown the design concentrations on these graphs as the horizontal yellow line. So compared to the design BOD of 75 milligrams per liter, the actual influent BOD averaged more than double that at 166 milligrams per liter, and sometimes reaching concentrations of over 300 milligrams per liter, as can be seen during this second half of the pilot, where BOD levels really started to spike. And then compared to the design of 15 ppm ammonia in the influent, we saw an average of about 24 ppm in the ammonia for the ammonia entering our system, which actually on its own, that ammonia difference isn't too bad, 15 to 24. Typically, our system would be able to, that's not an issue for our systems. But given what I've talked about in this webinar regarding the competition between heterotrophs and nitrifiers, you would anticipate that this discrepancy here would have a major impact on nitrification. And it kind of did. As you can see, our effluent ammonia levels are sort of fluctuating. But the key thing is that despite these BOD peaks and despite the cold weather temperatures, we actually still saw a lot of nitrification. And I want to show that more clearly in this table here. So on average, for the whole project period, we were able to reduce ammonia concentrations by 76%, or about 18 milligrams per liter in total that was removed from the influent on average. And that actually exceeds the design removals, since we planned for only 15 milligrams per liter of ammonia in the influent to be removed. So yeah, so we did see really great nitrification here. And we also saw that this nitrifier activity was sustained during cold weather conditions, where we actually saw better ammonia removals at an average of 80% ammonia reduction, despite water temperatures below 10 degrees Celsius, as well as an excessive BOD influent in the influent. So now that we were able to validate our ability to treat this wastewater, we were able to redesign and refine our full-scale design using more conservative design loading rates, one that was more reflective of the actual BOD and actual ammonia values that we saw during piloting. So we were able to kind of refine that design to account for all those fluctuations to make sure that we were meeting the targets that we wanted. So by doing this, we could ensure that we were getting the removals that we needed to consistently maintain an effluent ammonia that was below the discharge limit. And like I mentioned, I do talk more about piloting and how you can ensure your pilot is a success in next week's webinar. The next case study that I want to outline is from another cold weather ammonia removal project. This time, the facility in question was located in a city in central Western Canada. So this lagoon facility used 3 cells to treat a mix of industrial and domestic wastewater, with ammonia efficiencies being greatly reduced in the wintertime. So our client wanted to conduct a pilot to demonstrate BioCord's cold weather ammonia removal at their site. And specifically, they wanted to compare the cold weather performance of BioCord to the cold weather performance of their existing system, like without any technology upgrade to it. So we set up our containerized BioCord unit as a side stream process. And because we had limited historical data from this facility, we actually just picked some standard BOD and ammonia influent values and configured the pilot system to be able to add more reactors to the process if it was needed. But it didn't. So we started, which it did. So we ended up using 2 treatment tanks and 2 BioCord reactors with a total HRT of 22 hours to match the full scale design. So here are the results. Influent concentrations, again, are in red. BioCord effluent concentrations are in blue, and the dotted line is the temperature, which averaged around 7 degrees Celsius water temperature over the duration of the project. I also plotted the contaminant reductions as performed by just the existing cells, so without BioCord in it, and that's plotted in green for BOD or CBOD and in yellow for the ammonia graph. As you can see, the influent BOD remained pretty consistent, as did our BOD reductions, so we had no problems at all achieving those BOD discharge limits, which also allowed us to achieve our effluent ammonia targets. So after this initial acclimatization period, as nitrifiers are developing and reaching steady state, we were able to achieve the targets for ammonia reductions. So overall, these case studies that I have presented in this webinar were able to confirm the BioCord reactor's ability to remove BOD and ammonia from cold temperature lagoon waters. These results demonstrate a significant improvement in cold weather contaminant removals when compared to those observed when utilizing just the existing lagoon capacity alone, as per the industrial facility case study that I just presented. So you can see here, compared to the BioCord effluent, the ponds by themselves did not very successfully treat any ammonia and very minimal amounts of BOD, as would be anticipated for a conventional treatment system. Our results also help to validate that BioCord reactors may be used as a practical alternative to high cost and high footprint system upgrades. Additionally, the microclimates that can develop on a biofilm within the BioCord reactor can allow for insulation against cold weather effects on microbial activity. And this was evidenced by the continued gene expression of the nitrifiers in the winter months, as well as by the substantial reduction of organic matter and ammonia from the influent streams at water temperatures less than 5 degrees Celsius. And that's almost it. Before I end off and open the webinar to questions or comments, I just want to talk a bit about Bishop and who we are. What I'd like to highlight is our technology focus as a company. We've grown a lot over the past 20 years, and we've dedicated a great deal of effort into developing our technologies and systems to provide economic and environmental benefits to our clients as well as the surrounding communities. So we do this by designing systems that are reliable, easy to operate and maintain, and have low energy requirements, as well as require little infrastructure around them. Of course, we couldn't do this without a great team, and we're very proud of the dedication that our team shows in working collaboratively with each other as well as with our partners to continually improve the solutions that we offer. We have a number of different solutions that can be used separately or in tandem to address a whole array of wastewater treatment and solids management issues. I mentioned last week about the Bishop water solids management solution. These geotextile containers can be used not only for sludge and solids dewatering, but they can also be used for shoreline protection and prevention of shoreline erosion, as well as in permanent process facilities that require TSS removal or pretreatment. Our BioCord reactors, as I've covered, are used for biological nutrient removal and the enhancement of cold weather treatment. And we also offer a range of products that can help augment treatment or be integrated with other solutions to allow for turnkey or plug and play systems that can address more than one challenge at a time. So for example, our chemical conditioning and polymer injection systems are used in conjunction with our solids management solutions to effectively flocculate and help to separate solids from a liquid stream. Beneficial bacteria can also be used to help with bioaugmentation. So these are essentially blends of different type of wastewater treating bacteria that can be added to a system to help inoculate it or give it a little bit of a boost if you're struggling to maintain the population of a certain type of bacteria. And lastly, we also offer a rare earth coagulant for phosphorus removal called RE300. This is essentially a more effective, more versatile, and less hazardous alternative to chemical coagulants such as alum or ferric that is used to precipitate out phosphorus. So RA300 can be used pretty easily to help achieve really stringent phosphorus limits. And that's essentially it. So if you are interested in BioCord for your application, please feel free to contact either Roberta or myself. As well, if you'd like to receive updates or read about some of our past and ongoing projects, please don't forget to subscribe to our newsletter, which can be done by visiting the Bishop Water webpage. We will be sending out these slides as well as a little bit of literature to all of our attendees today, including PDFs of our general BioCord brochure, as well as the brochure for our containerized system. And I do want to mention that we have a technical results document that's available upon request if you're interested in using BioCord for a potential project and want to see a summary of results that we've obtained from some of our past projects. So that includes results from BOD, TSS, and ammonia removals, site descriptions, and overall performance as it relates to meeting the target limits. So if you want to get a hold of this document, please don't hesitate to contact myself or Roberta, and we can pass that along to you. So thank you everyone for attending. I'm glad that you were able to make it out today. I'll check on the questions. It looks like we've only got a couple, so luckily so I think we will be ending up early. But what is the largest full-scale installation in Canada in terms of wastewater flows and ammonia loads? So this would be the one that's upcoming in Limoges, Dundalk, Ontario. I've got a little cut sheet on that if you're interested, but it's basically a municipal installation that is receiving around 3,500 cubic meters of wastewater a day, and it's split into 2 cells. So that's the largest one that we will have in Canada.